Hello everyone. So today we are going to discuss about the viral genetics. So in the viral genetics, we'll discuss about recombination, reassortment, complementation, and phenotype mixing. Also, we'll talk about the genotype mixing. So first of all, let us talk about the recombination. So uh, I have dissipated the two viruses here. One virus with the black marker and another with the blue marker. So the capsid and the nucleic acid is denoted by the black color and another virus that is denoted by the blue color. So these two are the complete different viruses. So in the recombination, what happens is that there are two viruses. What happens is that there is exchange of genes between the two chromosomes by crossing over within the region of significant base sequence homology. That means the genetic recombination may occur when two different but related viruses infect a cell simultaneously. So these two viruses exchange the segment of the nucleic acid so that hybrid virus result. So you can see the hybrid virus that has the genes both of both first and second virus. So that so this um, so this hybrid virus has the genes of both the parents. Now let us discuss about the reassortment. So reassortment, when the virus with the segmented genomes, the segmented genome virus is example, we can say it is influenza virus. So when the viruses with segmented genomes exchange genetic materials. So for example, let us say the 2009 novel H1N1 influenza. So a pandemic emerged via the complex viral reassortment of the genes from the human, swine and avian virus, and it has potential to cause antigenic shift. So you can see there is one virus and it has another virus. So it has the hybrid capsid of the both first and second virus. Also the nucleic acid, it has both the nucleic acids of the first and second virus. So there is reassortment of the first and second virus. Now let us discuss about the complementation. So what happens is that there are two viruses. One is functional virus and another is non-functional virus. That means when one of two viruses that infect the cell has a mutation that result in the non-functional protein, the non-mutated virus complements the mutated one by making the functional protein that serves both viruses. For example, hepatitis D virus requires the presence of replicating hepatitis B virus to supply the hepatitis B surface antigen, the envelope protein for the hepatitis B virus. That means uh, if there is presence of the non-functional virus and there is one present functional virus, and by the process of the complementation, a functional virus is achieved. So it is a functional interaction between the genes product. So protein specified gene, you can say the gene product is the protein specified gene of the two viruses. One or both may be defective, resulting in the multiplication of one or both under condition in which replication would not ordinarily occur. So if there is present of all non-functional virus, so for example a person has only hepatitis D virus in their body, the virus will not replicate inside the body. But if the virus has hepatitis B, also hepatitis D, the hepatitis D virus requires the presence of replicating hepatitis B virus to supply the hepatitis B surface antigen, the envelope protein of the hepatitis D virus. So this is the complementation. Now let us talk about phenotype mixing. So phenotype mixing, what happens? There is virus A, virus B, which has the progeny 1 and ultimately the progeny 2. So what happens? It occurs with the simultaneous infection of the cell with the two viruses. For progeny 1, we can see genome of the virus A can be partially or completely coated, forming the pseudovirion with the surface protein of the virus B. So the type B protein codes determine the tropism, that means infectivity of the hybrid virus. So progeny from the subsequent infection of the cell by the progeny 1 will have a type A code that is encoded by the type A genetic material. So that means when two different viruses, virus A and virus B, multiply in a cell, so mix up may takes place during the assembly, so that the genome of the one virus can be surrounded by the capsids belonging to the partially or entirely to the other viruses. So let us talk in brief about the genotype mixing. So this genotype mixing is a heterozygous. This heterozygous result from the incorporation of one or more than one complete genome into the single virus particle. So this is all about the virus genetics. I hope you understood the recombination, reassortment, complementation and phenotype mixing. Thank you.